the electric transport system, we just started to talk about it. We're going to finish that up today, review that a little bit, make sure you understand what's happening, and then finish off the chapter. So that will give us time Wednesday to do a good review, and then remember the exam for Friday.
By the way, I bet there's probably a lot of things spelled wrong in this. 'Cause I
That's
do
okay.
it, like he says it and then I just write it down. So I never like actually know how things are spelled.
That's okay.
Except for like these I spelled right because I looked on the slides. But like
90% of the time, I can't spell worth a darn, so it's gonna be okay.
okay, cool. Like cytoplasm, I don't know if that's right. Mitochondria, I don't know.
It'll work.
Cellular respiration is a controlled reaction. Like you know what I mean though.
I can
Cellular
figure it out.
respiration.
I can figure out stuff real easy.
Just when he's like saying stuff, I'll just like write it down as fast as I can. So
That's why I do the videos, because
yeah,
I can't spell, and
write fast. Like
I can do it without looking at the
Okay.
paper at least. That's one plus.
that's talent.
Remember, glycolysis produces pyruvate from sugar. Pyruvate enters the citric acid cycle where electrons are harvested and electron carriers are formed, NADH, FADH2. Those electron carriers go to the electron transport system. In the electron transport system, which is what you see on the board here, you have got separation start to occur. Now, keep in mind, the electrons are down through here. The yellow is the electrons that you see right there. And the electrons are going to wind up in the cytoplasm, separated from the hydrogen protons. The only way for the hydrogen protons to get back into the system is back through the enzyme ATP synthase. Whenever the hydrogen comes through, the force that's created by the attraction between the positive charges and the negative charges is enough energy to make ATP. The hydrogen then comes back out and then can combine with oxygen to make water, where its electrons are.
The H pluses, do they come from when we have NADH and we have the plus H plus? So that's where the H plus comes from, right? Okay, so the electrons go on the chain and the H plus is pumped across the membrane.
Yeah, this NADH actually it's carrying the protons to a point. Notice right through here, it's got the NADH. Protons go up that way, and then the electrons come back out. It's separated. That's what the electron transport does. This is the electron transport. When the hydrogens come back through ATP synthase, that is chemiosmosis. Both of these together make the electron transport system. So basically, you have looked and learned, hopefully, down to the electron, what happens whenever we go through and start to take the energy out of food that we have. And there are some questions that you should always keep on your mind, like what are the products of those three stages? Difference between oxidative phosphorylation and substrate. Remember, oxidative is oxidizing, it gives up an electron. Substrate uses an enzyme to make ATP. You need to remember most of the ATP is made in the third stage, oxidative phosphorylation. When what we have looked at has been for glycolysis. Now the rest of the chapter, we're going to look at what happens with some of these other food molecules, what happens with protein, what happens with fats. So glucose is broken down to water and carbon dioxide. That's one of there's two main reasons why your body prefers to burn carbohydrates. Number one, they are simple to break down. And number two, the waste products produced are not toxic or harmful to us. They're rather benign, carbon dioxide and water. They actually create about 10% of the water a human needs in breaking down this food. So,
I'm sorry, how much, what did you say? How many?
10% about, and that is an estimate because different people's physiology is different depending on food. So. Notice once again up here, the energy stored in the hydrogen gradient. It's not saying the energy stored in hydrogen. Whenever you separate those charges, positive and negative, there's an attraction for them to want to come back together. When that is the gradient. Whenever that charge or the attraction gets great enough to pull them through the enzyme, then that's where that is jumped across the gradient. The gradient is how much of a separation there is between the charges in strength. So if we read this, it says the energy stored in the hydrogen proton gradient across the membrane couples the redox reactions of the electron transport chain to ATP synthesis. In other words, in the electron transport chains, we make the separation, build the gradient, and through ATP synthesis comes down through the enzyme ATP synthase, and then we have the energy made. The hydrogen gradient is referred to as a proton motive, which means to move, force. So the force of those protons moving to reunite with the electrons is called the proton motive force. That comes as a result of the gradient. Now, I want to caution you when we start to talk about how much ATP is produced. That is going to be dependent on the type of organism, on the type of cell, different things. What you are going to see with these numbers are an overall average with these things, not an exact number. Pretty close though. It says during cellular respiration, most energy flows in this sequence. These are the same answers that I say, what is the most important thing to come out of glucose? It's going to be pyruvate. From pyruvate, it goes into citric acid cycle, makes NADH. NADH is taken to the electron transport system, and we get ATP. Also, the actual correct name for those three stages are glycolysis, the citric acid cycle, and oxidative phosphorylation. Many people, including myself, often just calls the third stage electron transport system. You can break it down into the two parts, but it really doesn't matter. Also, 34% of the energy in a glucose molecule is transferred to ATP during cellular respiration. Now think about that. 34% of the energy contained in glucose is what we get out of it. We don't get near that much out of meat. Not nearly as efficient. Notice the term about 34%. Even in people, some people's bodies will be more efficient at extracting the energy than others. If this system, this process is fine-tuned, we're good. If it's not, we won't get as much energy. Notice it says we make about 32 ATP, and that's 32 ATP free energy for use. And as it says at the bottom, we can't tell you exactly how much ATP is made because people's physiology is different. If you give somebody one drug, one person's body could have one effect and another taking the very same drug could have a different effect. So oftentimes, like when doctors are prescribing things like blood pressure medicine, they know what should happen, but they don't know what's going to happen. So what they do is they may have to adjust the doses of things or even change drugs, depending on how an individual's physiology, how their body responds. And the same thing here. We're all don't carry the same efficiency as extracting that ATP out of food. One thing is known, and this is important to you. If you ever work in a healthcare field, if you've seen people, you ever seen somebody or even done it, you go out and you walk a mile and come back home, get on the scale, see how much weight you've lost none. Just because you walked a mile. Now you've used energy, but you've also had some byproducts with that energy. When you lose weight, what have you lost? What actually have you lost? Have you lost glucose?
Are you just giving off the products from the reactions of
You're
cellular?
losing the waste products. Now, where does the waste products come from? From glucose, carbon dioxide, and water. You don't lose that water immediately. Your kidneys will take out the excess water that your body has. You breathe out carbon dioxide. It takes a while before you start to see the effects of these things. It's not point. What we lose is going to be the waste products. Those molecules, carbohydrates, fats, and protein have been shifted into another form, another molecule. But there's something that's important. One of the more important things is being in shape. Doctors don't explain why. Something like it's important for weight loss. They really don't. If you ever work in the healthcare field, it is important for patients to understand what's happening. When you get your muscles tuned, in shape, they will be more efficient at extracting energy from these food molecules than if they were out of shape. When they become more efficient, little messages start to go to the brain, fuel the hunger complex. We don't need as much fuel coming in because it will last longer. So it actually will help to quell your appetite just by being in shape and becoming efficient. If, let's say your body is, as it says there, 34% efficient, what happens if you get it in shape and it goes up to 45% efficient? You don't need as much fuel, and your brain picks up on that. So what's important about exercise is not how much weight you immediately lose after walking a mile or whatever. It is getting those muscles in shape, the engine tuned, so that it needs less fuel. This is a rundown of cellular respiration. Notice, glycolysis will produce 2 NADH. Where does those NADH go? To oxidative phosphorylation. Also, there will be enough energy left over to make 2 ATP. Remember, NADH can make about 3, and once again, depends on the system, but 2 to 3. So the most important thing to come out of glucose or glycolysis is turning glucose into pyruvate, 2 molecules of it. Pyruvate will then be oxidized. In doing so, it will produce 2-NADH that will come into oxidative phosphorylation. Here, the pyruvate will be oxidized. It will lose a carbon dioxide, become just the acetyl group, a 2-carbon compound. That will then combine in the citric acid cycle with oxaloacetate to make the 6-carbon citric acid. Out of the citric acid cycle comes 6-NADH, 2-FADH that goes into oxidative phosphorylation. That's where those NADHs are going to be turned into ATP. It also, like glycolysis, will produce 2 ATP of energy. These 2 ATP out of glycolysis and the 2 out of the citric acid cycle are substrate phosphorylation driven by an enzyme. The other ATP that's made, as you see, out of oxidative phosphorylation, it says 26 or 28. 28, if you look at this, we've got 2 times 2. Remember, we've got 2 molecules of it. There's 24. All right, that's 4, and 4 is 8. And then if you get to 28 here, that's 36. 4 of those have to drive those 2 cycles, so that makes 32.
And you get the 26 or the 28 by multiplying. If FADH2 is about we said
About
2,
two.
and then NADH is either 2 to 3,
About
but we can say
3.
3. So then you multiply those out for each of
Well,
those.
if you look,
right?
you gotta remember, we've got 2 molecules of pyruvate. So that will that's 2 ATP net right there, right?
Right.
So here we're gonna have 4.
Okay. Write it down.
And the same thing in the citric acid cycle.
And does that also apply to the 2 NADH? Is that just representing for one molecule of pyruvate?
Pretty much, yeah,
Okay.
up there. All of these represent,
Just for one molecule?
yeah.
Okay.
If you look, just the one molecule, you've got 2 ATP in glycolysis, 2 out of citric acid cycle, so that's 4 molecules of ATP. And then if you go to the 28, that's about 32. So that's for one molecule. But now remember this, more than that's actually made.
All right.
Remember, in glycolysis, 4 ATP are made, but only 2 is free energy that you see down here. But 4 is made in glycolysis, then you've got 2 that's made in the citric acid cycle. That is 6, so if you add that to 28, then you get 34. That's what's total made, but 32 is how much free energy. So we have to get 2 of those back to compensate for the 2 we put in. And once again, remember, this is an average. And like they say, it's not the same. For bacteria are going to be different. Why would bacteria be different? Because
They use
pyruvate does not have to move across the mitochondrial membrane because bacteria don't have mitochondria. So it gets more net out than we do. So it says maximum for glucose, it says about 32. And it is dependent on the cells. This is the average. That is what to go by. Just go by the average in general. Some things will be more, some will be less, but that's the average. About 32 free energy ATP molecules.
So will you ask us this? I mean,
I will ask something along the lines, how much free energy do you get out of a glucose molecule? And that answer should be about 32. How many total ATP are made? About 34. But remember, 2 of those have to fuel the process of glycolysis. So free energy or net energy, only 32.
Oh, and then total would be 34.
Yeah, but I don't usually ask total, I just ask net energy to keep confusion.
Okay.
So the answer should be 32, is how much free energy that comes out of this process.
So I'm sorry.
respiration.
Free energy is the same thing as saying net energy, right? Because
Net
net
energy
is-
means after we paid the bill, how much do we have left over to use?
okay, because we already used 2 in glycolysis, so they're no longer usable.
Takes 2 to drive glycolysis.
Okay, thank you.
Now, what happens if and when we don't have oxygen? What happens if you run real hard? Think about what happens. Your breathing starts to increase. Why? You need more oxygen, right, to get to. When cellular respiration is going at one rate and suddenly it goes faster, you got to have more oxygen, so you got to breathe faster to get oxygen in. You've got to get that oxygen moved to the cells. So what has to do happen with your blood pressure? It has to increase. Your heart starts to beat faster then to make sure that you're pushing more blood to the cells. Once it drops off its oxygen and the glucose that your body needs to burn for energy, it has to get rid of the waste products. It has to move out things like that, carbon dioxide, any other nutrients. If you're just doing glycolysis or a glucose molecule, you'll get carbon dioxide, it'll be your waste product that's going out. What does that do to your blood? What happens if you put carbon dioxide into water, bubble it up? Becomes an acid, carbonic acid. So your body has to have a mechanism of producing a buffer to prevent that extra carbon dioxide from turning your blood acidic. And it does. It basically produces bicarbonate of soda. You've got chemical mechanisms in your body that will produce that to keep your blood pH at a specific level, because once you get beyond about 7.1 pH, you're in trouble. System shut down.
7.4 was the ideal pH for human blood.
Once again, it depends. What
Around
people-
7.2 to 7.4 is the range I read up.
That's within reason. So, if your blood pH gets out of whack, your other chemical reactions don't take place. Organs will start to shut down, systems start to shut down. Basically, your blood needs to be neutral.
Can you please repeat what that buffer's name was? The name of the buffer?
It's just bicarbonate
Oh,
of soda.
bicarbonate. Thank you.
That's what your body produces. It's a base and alkaline, and it lowers, it works against a low pH, it will raise it.
Okay.
Remember, the definition of a buffer is to resist a change in pH, so it wants it to stay at what's normal. And folks, once again, with these numbers, what's normal? What's normal blood pressure? I mean, temperature,
98.
98.6. A lot of people out there is 97, does just fine, I am. That, once again, is an average. There are low numbers, and there are people whose body temperatures will be slightly higher, and they're perfectly normal. The 98.6 is when you take everybody and just come up with an average. Same way with blood pressure. Same way with pulse, respiration. So the things you're getting are averages. Okay? Now, if you don't have oxygen going into your cells, muscle cells, at the rate the muscle cells are using energy and producing the waste products. In other words, your body's demand for oxygen is greater than what your body can supply, then cellular respiration shuts down. It cannot operate without oxygen. So you have a limit. You can raise that limit if you exercise and get into shape. The more demand that you put on your muscles, the body will eventually grow little microscopic blood vessels to increase the amount of oxygen that it can bring to the cells and the amount of waste that it can remove. That's what all that getting in shape is, just stressing yourself. Now, what happens once you reach that limit? You're running and you can't do any more, you don't get enough oxygen to keep the system going. Then your body goes into fermentation, anaerobic respiration. Glycolysis can still continue. It doesn't need oxygen. So while you're getting tired and running down, glycolysis is still occurring, but you're only getting 2 ATP of energy out of it, which is nothing for your body. Then the pyruvate can't go any further. It builds up. So your body has devised a way to get rid of that pyruvate, turn it into lactic acid. By turning it into lactic acid, it can move out of the cells. It can go into your blood, through your urinary system. You can get rid of lactic acid. Muscle cells can store a certain amount of it. And if you ever go to a gym and you work out and then you come out with your muscles feeling swell, that's because of the lactic acid that's built up in it. It will go away. Wait 2 days and your arms don't feel nearly as big as they did because the lactic acid is gone. Your body muscles at a later time can use that lactic acid. It can reverse itself and go then back through cellular respiration, but it can only store so much. Notice, as it says up here, most cellular respiration requires oxygen. Without O2, the electron transport chain stops, but glycolysis can continue. That is anaerobic, no oxygen needed. Anaerobic uses an electron transport chain with a final electron acceptor other than oxygen. For example, sulfate. Fermentation uses substrate level phosphorylation instead of the electron transport chain. Now, when you look up that first line, that is not necessarily talking about people. That's talking about, it is in a way, but there are some bacteria, for example, that do anaerobic respiration. That's the only kind they do. They found a way to use something besides oxygen as the final electron acceptor. Bacteria can use some of these different things. That is anaerobic respiration. Now, when we stop, glycolysis produces pyruvate. Then it goes into our pathway, which is fermentation. Fermentation consists of glycolysis plus reactions that regenerate NAD+, which can be reused by glycolysis. Notice that's not energy, that is NAD+. There are 2 pathways from fermentation in organisms, 2 products. Basically, alcohol or lactic acid. Alcohol will also produce carbon dioxide. When we go through, you make bread, and the yeast goes into the bread and causes the dough to rise. The little bubbles created are carbon dioxide bubbles that get into the dough to cause the fluffiness. With yeast, they can also go into alcohol fermentation. If you do both of these Certain yeast will produce more carbon dioxide, a little bit of alcohol, not much. Bread has got that smell like a beer baking when you get into it because some of the byproducts are the same. But there's extremely little, if any, like 0.1% alcohol produced. So it's not very much at all from the type of yeast that make bread. There are other type of yeast called beer yeast. Now they will produce maybe 8 to 9% alcohol whenever they go through fermentation. Your mussels can go through lactic acid fermentation. In alcohol fermentation, pyruvate is converted to ethanol in 2 steps. The first, once again, releases carbon dioxide. Alcohol fermentation by yeast is used in brewing, winemaking, and also in baking. It's just, there's a difference in the yeast on how much alcohol they produced. This shows the process. You don't have to remember that. Notice up here, pyruvate gives off this carbon dioxide, and we make a little molecule similar to the acetyl group, but we've added a hydrogen to it. Now it is called an acetaldehyde. You look right over here. That part's the acetyl group. Now you add a hydrogen to it and it becomes acetaldehyde. The acetaldehyde will then be converted to ethanol. Remember, basically, the ethanol, methane is CH4. Ethane, or ethyl, is 6. So, 1, 2, 3, 4. These bonds take the place of that. This is the ethyl group. 3, 4, 5, 6. So when we take that acetaldehyde, notice we got 2 carbons, 1 oxygen, and 4 hydrogens in the acetaldehyde. We had a couple hydrogens. Now we got ethanol. Hydroxide, remember, makes an alcohol when it's added. You don't have to remember that, but I'm just trying to show you. Glucose turns it into pyruvate. Pyruvate will take the carbon dioxide off like we've got there to make the acetyl group. But that acetyl group then goes into citric acid cycle to combine with oxaloacetate. Here, when the carbon dioxide is lost, the hydrogen is added. No coenzyme A. The hydrogen is added. We've got 4 hydrogens, 2 carbons, and oxygen that go. We had hydroxide group. Now we've got ethanol.
Wait, I'm sorry, where does the hydrogen come from again?
It's coming out of the system and through there. The NADH. NADH uses the hydrogen. That's how the NAD is regenerated. In
Okay.
other words, the NADH is not going to make energy. It's serving as a source of
Oh,
hydrogen.
right.
And what energy it's got helps to fuel that reaction.
I mean, like the hydrogen over on the right, when it's coming out of the pyruvate and it releases the carbon dioxide and it has that.
It's all coming out of the NADH.
Oh, okay, it's still coming out of the NADH. Okay.
All of that in through there, and then this. Notice how it's going in this direction. It comes around. That's what we're just trying to say.
Okay.
I don't want you trying to remember this. I just want you to understand there is a process similar to glycolysis of citric acid cycle, but it's a different pathway. We don't use the coenzyme A. Stick a hydrogen on it, add it to a methyl or ethyl group, and you got alcohol. So that's their final product.
Okay.
Notice it also produces carbon dioxide, right? If you've ever seen anybody making wine or beer when you first started, all that sugar in there is being fermented and broken down, produces a lot of carbon dioxide coming out. In things like beer making and wine making, you really don't want this to be open to the air. So they use the little cap locks that will go down in top of the container that will let the carbon dioxide bubble out. They're trying to prevent something from happening. Anybody know what it is?
I know that a lot of bacteria.
There's a type of a bacteria called Acetobacter. And if they get into vat of wine or beer or whatever, they will turn the alcohol into vinegar. That is the way we get vinegar. You take wine, white wine, put that acetobacter in it, let them convert the alcohol to acetic acid, then you distill it and remove the junk part, and that's distilled white vinegar. If you do that with grape or red wine, you get balsamic vinegar. And they will actually put that into big containers and let it evaporate out, get the moisture out, and almost get thick, and then it becomes extremely sweet, and actually use it for ice cream topping. Balsamic vinegar, aged. So that is all because of alcohol being converted by the acetobacter into vinegar.
I've heard about that vinegar. It becomes thick like a syrup, and they can put it on top of like desserts.
It's sweet. It's kind of like sweet and sour.
Right. I think it was on like a TV show on Netflix.
The
like
older that it is, the more aged, the thicker it gets, and more expensive it gets. I mean, some of that can cost as much as wine.
Wow.
It's aged in barrels and things for dozens of years sometimes. And the barrels actually absorb some of the water out, and that makes it thick enough. And it can be used completely for ice cream toppings, syrup. In lactic acid fermentation, pyruvate is reduced to NADH, forming lactate as an end product, with no release of carbon dioxide. That's a key thing. Lactic acid fermentation don't produce carbon dioxide. If you go home, take you a quart of milk, you can heat it up just to where it starts to look a little bit frothy, but not boiling by any stretch. Warm it up, take it out, and then set it on the counter and let it cool. Go in to your refrigerator, whatever. If you got yogurt that says active cultured yogurt, take just a spoonful, put it in that quart jar, give it 2 days, you've got a whole quart jar then of yogurt. Completely natural, no sugar. It won't taste like any yogurt you've eaten because it has nothing in it. It's just the lactic acid. And then people will often put things like berries, blueberries, or sugar to it, and you can make yogurt that way and just keep it coming. And that's similar to the way buttermilk's made.
What does the warming of the milk initially do? If you let it cool down, what does it do?
Proteins.
Oh,
You're
it did.
starting to not completely to nature, but breaking it down.
Oh, I see.
And then you can take plain dough, just plain dough, add water to it, set it out on your counter for 2 or 3 days, and you basically got sourdough. When you come back, depending on where you live, the fungus, the yeast cells that's in the air will turn it into sourdough. The reason places like San Francisco and Alaska, both on the West Coast, are known for sourdough bread is because they have the type of fungus there, the yeast, that will give it that real sour lactic acid flavor. Lactic acid is what gives the sharpness to things like yogurt and so forth. Notice here it says lactic acid fermentation by some fungi and bacteria is used to make cheese and yogurt. Human muscle cells use lactic acid fermentation to generate ATP when oxygen is scarce. Now, I can make some. They don't make enough for you to need. Comparing fermentation with anaerobic respiration, glycolysis is 2. In all three, NAD positive is the oxidizing agent that accepts electrons during glycolysis process. I don't really want you to remember all of that much, just remember the products. Is fermentation is going to be alcohol and carbon dioxide. In muscle cells, the product will be lactic acid, and it does not produce carbon dioxide. Notice fermentation produces 2 ATP per glucose, whereas cellular respiration 32. So cellular or fermentation just does not produce enough. I want you to know these terms. Obligated to. Obligate anaerobes carry out fermentation or anaerobic respiration, cannot survive in the presence of oxygen. These cannot be in oxygen in function. Most of the way they survive in nature is they will roll up into a little ball called a spore, and they will wait until the conditions get right, and then they will vegetate or grow back into the bacteria. They are anaerobic. They cannot survive in oxygen in function. Yeast and many bacteria are called facultative. That means they can either go through fermentation and do fine, aerobic, or they can either do aerobic respiration or anaerobic, either way. Oxygen, they can use it, they can do without it. So it doesn't matter. It doesn't kill them. It says they can survive using either fermentation or cellular respiration. They can do that because they are small and they don't have a large requirement of ATP. And facultative, pyruvate is a fork in the metabolic road that leads to alternative catabolic routes. And remember the old saying, if you ever come to a fork in the road, take it. Glucose comes into the cell. It's going to make pyruvate. If oxygen is present, it goes through cellular respiration. If oxygen is not present, it goes in through, if it's in people, lactic acid, fermentation. If it's yeast, ethanol, could be lactic acid if you're bacteria. Yep.
So, facultative,
They can live in the
anaerobes.
present and do it or live without it.
Is that the name of the cell? The cell is
No,
considered,
it's type of organism, a facultative organism.
okay.
And most of these organisms are one cell. Bacteria, primarily. Notice, we've been talking about glucose and glycolysis. The definition of glycolysis is the breakdown of glucose. There are other molecules that we need to break down, such as fats and proteins. Glycolysis and the citric acid cycle are the major intersections to various catabolic and anabolic pathways. Catabolic pathways funnel electrons from different organic molecules. Remember, catabolic means to break down, to release energy. Proteins must be digested to amino acids. Amino groups can feed glycolysis of the citric acid cycle. But there's a problem. Remember how if we go through glycolysis, we get 34% efficiency getting in, not even close with protein. I mean, it's a minute amount. If we tried to survive totally on proteins, we would not. We couldn't do it. Remember, proteins must be broken down into amino acids, and then the amino groups can go into glycolysis. Look at fats. Digested into glycerol. What is glycogen? That's animal storage carbohydrate form, right? The storage, glycerol here is basically an oil, glycerin. Fats are digested to glycerol used to end glycolysis, so it can go into the system similar to where glucose would. It's used in regenerating the acetyl-CoA, like in glycolysis. These fatty acids are broken down by beta oxidation. Basically, that's the type of bonds that are broken. You don't really need to remember beta oxidation. Remember, the fatty acids are broken down to acetyl-CoA in glycolysis. They are broken down to acetyl-CoA. An oxidized gram of fat produces more than twice as much ATP as an oxidized gram of a carbohydrate. Fats contain more energy, but they are more difficult to break down and produce not as good waste products, not as non-toxic. And notice this little thing right here. This is glycolysis. This is what happens with carbohydrates. Notice fats come in at that G3P stage. Remember, that was at the end of the investment phase. Fatty acids come in and are just converted at the end to acetyl-CoA, so they're not going through this process. Then that acetyl-CoA can go through the citric acid cycle and oxidative phosphorylation like before. So even though carbohydrates contain less total energy, we get more out of it. Remember, we're not getting that ATP that we would normally get out of glycolysis. It's coming in around. Glycogen can, or glycerol can give us some. What role does glycerol play in a fatty acid? Remember, glycerol is what holds the fatty acid chains together. It's on a fatty acid chain. So you break off the glycerol, it goes in one pathway, and the fatty acid then goes down and is turned into acetyl-CoA. Now here's the key on amino acids. Look where they come in. They likewise can be converted to acetyl-CoA. But we have to go through a process called deamination. We have to get rid of the amine, the amino group. We lose a lot of energy right there. Many amino acids don't even come in to the citric acid cycle until right there. So all of that NADH and all the FADH that's made before that, we don't get. We get one little bit of NAD made at the end, the last quarter. So even though there is energy in protein, we get very little out. That's why some of these all-protein diets do work before they kill you with liver failure, kidney failure, because of the ketones they produce as a waste product. So your body prefers carbohydrates, and it prefers them for a reason. That's about 12:10. We'll pick up Wednesday.